Vedi? Una di 17.
No, questa è la presentazione.
Ah sì, ok.
Sì, infatti dove...
Hereby, I open this academic ceremony in which Giorgia d'Italia will defend the academic thesis From Platelets to Long Non-Coding RNAs, Understanding Heart Failure with Preserved Ejection Fraction. May I invite you to present a summary of your studies and the conclusions of your thesis.
Dear Pro-Rector, dear member of Corona, dear family, friends, and colleagues, in the next 15 minutes, I would like to give you a brief overview of the main aim, result, and conclusion of my thesis, the research I conducted in the past 4 years, and I hope to get you a little bit into the fascinating world of platelets and non-coding RNA in the context of cardiovascular disease. Do you know that blood is more than a red liquid? Well, actually, it's a really important part of our body. And it is complex because it's made of different components, including platelets. But what are platelets? Well, platelets are small but essential cells in our body. And maybe you know them because are the cells that help us to stop bleeding. In particular, when you get cut, platelets start rapidly to get activated and to stick to the damaged area and to each other. And together with the coagulation system, they can form a stable blood clot that can help stop bleeding in a process called hemostasis. But if platelets get overactivated, they can form some unwanted and dangerous blood clots that can occlude the vessels in a process called thrombosis. And this can lead to serious consequences like heart attack or stroke. So platelets are small, but really important in these processes. However, platelets can do much more than this. Indeed, platelets are pretty social cells because they can interact with other components in the blood. For example, they can interact with the cells of the immune system, so the cells that help us fighting against bacteria and virus infection. And they can also interact with the cells that form our blood vessels. And they can do that through a direct interaction or by releasing signaling molecules that can help this communication. So studying all these mechanisms, scientists found that platelets actually are involved in a number of different functions, and some of these are now emerging as really important. like, for example, the platelet role in the immune response, but also in inflammation, which is the first reaction of the body after an injury, and also how the blood vessel function. And in all this function, platelet can act both as good and bad players. And this is important because all these platelet emerging function are now seems to be important in disease conditions that are essential in cardiovascular context, like high blood pressure and diabetes. All these mechanisms are also emerging as important in a syndrome called heart failure with preserved ejection fraction, HFpEF. Patients with HFpEF often show serious symptoms like shortness of breath, fatigue, and weakness. Moreover, these patients are often of old age and mainly females, and they also often present other cardiovascular and non-cardiovascular conditions. But what is happening in these patients? Well, actually, compared to a normal healthy heart that is able to relax and fill with blood easily during the heart pumping, in HFpEF, the heart muscle became stiff, so it's not able to fill properly anymore. And heart failure is indeed a really important and serious clinical problem, because these patients are really sick patients, but actually we still don't know what are the causes that lead to the disease, and there's also a lack of a specific targeted treatment. And this highlights why it's so important, the research in this context, and the need to better elucidate causes and to find novel targets for medications. So finding novel targets for medication is an important area in HFpEF research and can involve special molecules called non-coding RNA. One of the main scientific results of the early centuries was decoding the human genome, so our DNA. And what surprised the scientists most is that actually only a small percentage of the genome actually code for functional proteins, so the functional building blocks of the cells, while all the other part is a non-coding part. And for a long time, this non-coding part was considered as junk or trash DNA, so not important for us. But actually, this is not true, because this part can produce some molecules called non-coding RNAs. Non-coding RNAs can be of different sizes. They can be small, the so-called microRNAs, or they can be longer, the so-called non-coding RNAs. These molecules are important because even if they don't produce a functional building blocks of the cells, they can regulate gene activity, so when the genes are on or off. And they are becoming quite a hot topic in the cardiovascular field because of their emerging role in the heart and their potential as therapeutic targets. So HFpEF, it's a really complicated and heterogeneous syndrome, and all the mechanisms that lead to the disease and the causes, the so-called pathophysiology, is still partially unknown. In my thesis, I elucidate part of the HFpEF pathophysiology by, on one hand, investigating the role of circulating elements like platelets, and on the other hand, investigate the role of long non-coding RNAs. So I dive a little bit into the understanding more HFpEF through the different chapters of my thesis. And in particular, in chapter 2, I review the scientific literature about platelet function in HFpEF and in the related conditions. Then, in chapter 3, I actually measure platelets, coagulation, and neutrophil function in HFpEF. Then, in chapter 4, I investigated whether platelet feature were linked to markers of disease progression. Then, in chapter 5, I investigated a special long non-coding RNA, NIT1, and its role in mouse heart under stress. Then, in chapter 6 and 7, we bring all these findings together. We discuss their meaning based on scientific literature and their impact on society. So, as I mentioned before, HFpEF is often linked to other conditions, like, for example, high blood pressure and diabetes. And in chapter 2 of my thesis, I revised the scientific literature about platelet function in all these different conditions and in FF itself. And from our analysis revealed that actually there are many commonalities among all these conditions, and particular compared to a normal functional protein, platelets, platelets in most of these conditions looks aberrant. And this means that have a difference in the way that get activated, in their size, and in the molecules they release. And this can affect the way they communicate with the surroundings. But what about platelet function in FF itself? Well, actually, few studies investigate this aspect so far, and there are still many open questions in the field. In chapter 2, we also identify novel and emerging research topics in platelet biology in the context of FF. In particular, the emerging idea that platelets are more complex than what we thought before, and they are all different, and they can see as homogeneous population, but they can have different characteristics. Moreover, also the increasing interest in studying the difference between females and males during aging, an aspect that for a long time was understudied in both in medicine and biology, but also focusing on the interaction between platelets and the components of the blood vessels and their communications. In this chapter, we indeed highlight the knowledge gap in the field related to platelet biology and FF. And try to fill this gap. In chapter 3 and 4, I studied the role of circulating elements like platelets in FF with patients' material. In particular, I collected blood from more than 100 patients and from 40 controls. And with this blood, I performed different experiments and analyses to indeed investigate the role of these elements in FF. In particular, I investigate the platelets, neutrophil coagulation, and endothelium activation marker in the plasma. Then I also check for platelet reactivity. It means how platelets get activated after an external stimulus, and also how they form the thrombi. And also perform some statistics to check whether our measurements were linked to FF and to markers of disease progression. And our experiments show that actually platelets derived from FF patients have different phenotype and characteristic compared to the control ones. Like, for example, the thrombi formed from the blood from FF patients were smaller, less contracted, and less active compared to the control one. Overall, we observe an aberrant platelet phenotype in FF patients. And of interest, this platelet dysfunctionality were linked to FF and not to the related conditions or medication use. Moreover, we also link aberrant platelets to some markers of disease progression. Moreover, we also found neutrophils and coagulation activation in plasma-formed patients compared to controls. Overall, in chapter 3 and 4, we identify platelets as novel players and potential targets in FF. In particular, we identify an aberrant platelet phenotype linked to FF, and we also identify platelets as potential marker to help predict disease progression. And this is important because I had knowledge to the complex pathophysiology of FF. However, we still don't know if platelets played a good or a bad role in this context. To further elucidate FF pathophysiology, we focus on the role of the special molecules, the non-coding RNA. In particular, in chapter 3, we focus on a specific one, the so-called NIT1. NIT1, together with other proteins, form these special structures called paraspectral that are inside the nucleus of cells. These structures are important because are involved in gene activity regulation and in the way the cell responds to stress. In chapter 6, we investigate whether eliminating NIT1 can make the heart failure worse. And to study that, we use different mice models and in different conditions. In particular, we have the normal control mice where NIT1 was normally expressed, and then we have the mouse where NIT1 was off, the so-called knockout. And then we also have the control heart and the one where we induce heart failure. So when you induce heart failure to a normal heart when NIT1 is on, normally the heart became enlarged and damaged. Interesting, our results reveal that when NIT1 is deleted, the heart is actually protected from failing. And this means that the heart is less sick, it has a better function, less scarring, the so-called fibrosis, and less inflammation. So in chapter 5, we identify NIT1 as an important player in this context. Finally, the main conclusion of my thesis is that we identified platelets aberrant phenotype in FPEF and in associated conditions. We also identified platelets as a potential marker of disease progression, and we propose NIT1 role in cardiac stress responses that lead to heart failure. Overall, I think that with this thesis, we've made a step forward in understanding the complex pathophysiology of FPEF. And with that, I would like to thank you all for your attention, and I would like to give the word back to the prorector.
Thank you very much for your presentation. It's now time to start the opposition, and the opposition will be opened by the chair of the assessment committee, Professor Henskens, Professor of Clinical Chemistry at Maastricht University Medical Center.
Thank you very much. Dear candidate, first I would like to congratulate you on this achievement. It's a very interesting topic with important results. And I like platelet function tests, so you have a lot of them in your thesis. Of course, I also want to include your supervisors, your family and friends in these congratulations. And also I like the cover because I see a sort of patchwork heart. I don't know where the platelets are, but it shows that you are a very creative person and that is really needed in research. So I want to ask my first question on your choice of 3 platelet activity tests. You measure activation markers in plasma. You do a flow cytometry test, which is a static test, and a flow-based model. What was your hypothesis to choose these 3 tests for your research? Because it's a lot of work.
Yeah. So, alias team opponents, thank you for your question and your compliments. So, yes, indeed, was quite a lot of work. I think the main aim was, okay, we received 6 ml blood from patients and controls, and we want to get as much as information we can about platelets. And we know that platelets are really complex, and they have different pathways of activation, and you can test this through different assays. So we believe that have a sort of heterogeneous type of assays can give a sort of more a clear overview of platelet function. Okay. And this is why we add the different type of assays.
But now when you look at the results, and then maybe that's really easy, but I looked at, for example, the summary at the end, and then you say, well, the flow cytometry shows some impaired activation and granular release. Then the thrombus formation, the flow test, shows also diminished. But the markers, they are the same in your control group and in your patient group. If you look back at it, what would you do now for a next study? Would you do all the 3 tests again? What information did it give you?
Yeah, honestly, I will repeat that because I think it's an important information to, because I think that this adds something to my results and not the opposite. I mean, of course, they can seem contrasting to maybe, but I think that add information because, for example, if I will choose to just measure the platelet function through the markers in plasma, I will receive an information just on the systemic in vivo activation, and not in the way they need form thrombi, or they express receptors, or they secrete. So I think this had something, so I will repeat that and even measure more.
Okay, measure more. Okay, but then I want to go to table 1, your baseline characteristics on page 54, because then I really want to understand, but I see that your control group has less anticoagulants. I see that the difference is between your on page 54, your patients, they have 50% they use anticoagulant drugs, so to diminish fibrin formation, and your control group only has 15%. And the percentage of platelet inhibitors is almost the same in both groups. No. How can I see that back in your results? Because is it, for example, on page 59, I can understand, for example, in figure E, that the fibrin formation is less in the patient group because they are anticoagulated.
Yeah, but we exclude actually the anticoagulant user from this analysis.
Okay, and so so you excluded the anticoagulant users, but not the platelet inhibitors.
No, not the platelet. No, exactly. Okay. Only the anticoagulants, because we do see that actually this can correlate with fibrin formation. So we decided to exclude that. But actually, we think that doesn't have an effect on other platelet measurements.
Well, that's really interesting information, I think, because how can you explain that the patients who don't use the anticoagulants form really, because if I look at figure E, they really have less fibrin formation.
Yeah.
And I always thought that all these people had a lot of thrombin generation. And what do you think is the clue there?
Yeah, I think that this is linked to the fact that, in general, the thrombi from these patients are way less, in general, less active, less contracted, and also the platelets are less active there. And so I think it's linked to the general platelet phenotype of these patients.
And if you conclude at the end, can I ask one more question? If you conclude in the end, well, it's not so correlated to medication, and both groups use platelet inhibitors, then what do you think is the cause then? Because the platelet inhibitor use is not different in these two groups. Are these platelets maybe already more exhausted? Or what's happening with these platelets?
I think so. I think it is more an intrinsic characteristic of the platelets from these patients compared to the controls ones, due to FF, so that's the presence of FF.
Okay, and we know that in your model also von Willebrand factor might have an influence. You didn't measure von Willebrand factor.
No.
Was that on purpose, or was there not enough blood anymore? Of course, that could also be the reason.
We overlooked this actually, but it would be interesting to do.
Okay. Do I still have time?
Half a minute.
Half a minute.
So.
Okay. Maybe one last question. I'm curious about, because we also developed a flow chamber for 37 degrees. Were your experiments with the room temperature flow chamber? And could you imagine what would happen if you would use the 37-degree flow chamber?
Yes. So in my setup, I use indeed room temperature and in under coagulation condition. And I think that at 37 degrees, I will probably see the effect of medication more. Probably.
Yes. Thank you. I agree with you. Yeah. Okay. I give the word back to the poet.
Thank you, Professor Henskens. The opposition will be continued by Dr. La Konen, Associate Professor of Translational Physical Activity and Health from the University of Jyväskylä in Finland, and she's present online. Dr. La Konen.
Thank you. Let's start with a general question. This is very extensive and impressive thesis, I would say, that because it includes one large literature review, which is already published, and then 3 experimental works. So now when you look at your studies and works, which of them makes you most proud of? And is there, on the other hand, in some works, did you feel that you needed to make compromises? And that is very common in academia due to the lack of time or financial resources or other resources. But if you now would think about them doing them again, is there something that you would do differently if you wouldn't have any restrictions?
Esteemed opponent, thank you for your questions and your compliments. I mean, I think that so this was the one of the first time that in our lab we'd really start measuring platelet function in such a large cohort. So I think that was the first experiment actually. I think it went well, but of course there is room for improvement. I think that with the best would be to obtain more blood from these patients and try to perform additional measurements and focus also on other cells in the bloodstream. That's something that I will do for sure. Another option could be also to for example, measure p-selectin expression also on the FACS, for example, and not only on the microfluidics. That can add some information. So yeah, overall, I think that we did a lot with the amount of blood that we receive, but I think that you can still do more if you get more blood and and in general,
yeah, great response. Yeah. Okay, let's go next to the chapter 5. That is also very impressive work, quite sophisticated experimental animal models that you used there. You are shared first order in this work. Can you explain what is your personal role in the work?
Yes, of course. So, indeed, this process was started before from another PhD student, and she did the initial part of the project where they collect sample from the animals and they start looking to the characteristics of the heart and all the different functional measurements. My contribution were more in the RNA and in the protein part, in the measurement, and also in the statistics part, and in the rewriting of the manuscript with additional results that we got. So yeah, that was my contribution to that. And yeah, indeed, it was a quite big project that takes quite some time to be developed and to end up to this full chapter. But yeah, that was my contribution.
Yes, thanks for the clarification. It was great that also this work included male and female animals. But then I got a little bit confused about the wordings that were used. For example, the gender and mixed gender, because I would have used personally the word sex because they are animals. Another thing that got me a little bit confused was that you didn't really report the results by the sex. So I asked the candidate a little bit elaborate about these wording choices and if there's possible influences of the sex on the results, and how could you test that?
Excuse me, could you repeat, please, the last part of the question?
The possible influence of the sex or gender, if you prefer that, on your results, and if that would have been possible to report separately, or how could you test the influence?
Yeah, so in this part is is missing for the chapter. I think that's probably with an integration separately will be a better choice for that. That's.
Okay, thank you. I think that my time has been up already. Is it?
You still have a minute or so, so if you have a short question, you can ask it, and otherwise we'll continue.
Okay, very shortly about the role of the non-coding RNAs in platelets, because in the chapter 2, a little bit also in the chapter 5, you indicate that the platelet-derived non-coding RNAs might have regulatory role. But to understand that, we need to understand where they come from, because the platelets don't have anymore the nucleus. So can you elaborate this a little bit?
So indeed, so platelets, platelets are one of the main sources of microRNAs actually in circulation. So they have a large amount of different microRNAs that have an important function. And actually it's becoming way more hot topic in the field to understand this field in particular because of the communication between platelets and endothelial cells, for example, and also because these messenger microRNAs are released in circulation and they can be uptaken from other cells, and this can then lead to the gene regulation on the uptaken cells. So it's really becoming an important topic in the field and in both disease and physiological conditions. And so I think it's a really important topic, non-coding RNAs, specifically microRNAs in platelets.
Yes, thank you.
Thank you, Dr. Laakkonen. The opposition will be continued by Dr. Giudetti, who is also present online. He's Associate Professor of Platelet Biology at the University of Pavia in Italy. The floor is yours.
Thank you very much. Thank you very much. My questions will be mostly about the results presented in Chapter 3 and platelet function in general. The study was certainly very impressive, and the availability of samples from patients is certainly a plus for any research. I was wondering whether there are reliable mouse models that could be used to study platelet function a little deeper to access to, let's say, more technical approaches.
Esteemed opponent, thank you for your question and your compliments. In particular, in the case of FPEF, the lack of a good preclinical model it's an important point. I mean, there is debate in the field because you can study different aspects of the disease development. Like, for example, in the chapter 5, we use attack mouse models, but was more linked to cardiac hypertrophy. Actually, there is now emerging a mouse model where you have on one side the more metabolic dysfunctionality, so link more on high-fat diet, and the other part is more linked to the nitric oxygen functions. So the creation of these models that have like multiple level of the disease, because the real difficulties of the FPEF is that it's a complicated disease, and it's often linked to other conditions that really it's believed that are part of the pathophysiology. So to create a model that can represent all this complexity is not easy, but it's now developing.
Okay, I have another question that is now related to the platelets from HPEF patients. I was wondering whether in literature there is something available about the ultrastructure, so for instance, electron microscopy analysis, to understand whether the morphology of these platelets is altered in addition to their function?
That's a really important point because, like in general, size it's important because some of the studies, for example, linked the platelet size to the worse outcome in the patients. Actually, we did measure just the MPV in these patients, and we didn't find any difference between the patients and the control. But that was more quantitative measurements of the size of the platelets. It would be really interesting also to dive a little bit more on the structure of the platelets, also linked to, for example, the secretion ability, and also we know that size is also linked to, of course, the function, and also to the idea that possibly larger platelets can have higher expression of receptors on the surface, maybe more granules, and this can influence, of course, the communication with the surroundings and their active action. Okay. So that part would be, and it's really, really interesting to dive into.
Yeah, I was actually thinking about the presence of granules, different numbers and size of granules, so it is connected to that. I go back to a question that was asked previously, actually not the same question, but the same, let's say, context. I was impressed too by the large number of investigations performed on a very little amount of blood that, of course, you obtained from patients. I was wondering, since you studied platelet activation and neutrophil activation separately, whether it was considered during this study to also study the formation of platelet neutrophil aggregates, which is, let's say, relatively simple technique that uses only a little amount of blood. I wonder if you considered that, or if it would have been a good idea.
Yeah, absolutely. It is a good idea, and because actually it also came during the process, because at the beginning we start with measuring platelet function, and then we did find this difference between patients and controls. So we then wanted to measure also in vivo activation, and then we say, okay, but platelets, they are not alone in the circulation, so let's see if also neutrophil or coagulation is also altered, and then also finding this increased neutrophil markers in plasma. Yeah, that's leads to the next question. Okay, how is the interaction between platelet and neutrophil, and is the formation of this aggregate increase or decrease in the patient? That will be for sure things that I would have done if I would do the study again. That's
okay. I hope I have time for one short question.
I'm afraid your time is up.
No problem. No problem. Okay. Thank you
very much. The opposition will be continued by Professor van het Hof, Professor of Intervention Cardiology at Maastricht University Medical Center. Also.
Yeah, thank you very much, Pro-Rector. Congratulate candidate. It's really amazing work on this difficult to study topic. As an interventional cardiologist, I'm very much interested in antiplatelet therapy. So that's why I asked one of your paranymph to read statement number 5.
Targeting platelet dysfunction represents a promising therapeutic strategy for FPF. This thesis.
Thank you. Thank you very much. Yes, I would like to ask you can you elaborate a bit on this statement, dear candidate?
Yes, highly esteemed opponents, thank you for your compliments and your question. Yes, of course. In these studies, we found that platelets are dysfunctional, and they look aberrant, and their function is different from the control ones. FPAF, I think it's the first time that there was like really a light in the platelets in the world of FPAF, because in general it's not like it wasn't really emerging the importance of platelets in this context, and I think that this gives a nice opportunity to think if targeting this platelet could be an option. Of course, we still didn't really went into the mechanism and why these platelets are dysfunctional. And also we know that for, in general, these patients are, they have like other comorbidities that can lead to the treatment with anti-platelet medications. But I think that...
Yeah, sorry to interrupt. I just want to know, because you mentioned on page 203 that this aberrant phenotype is either a cause or the consequence of the disease. So what do you think? Is it a cause, this aberrant phenotype, or the consequence?
Well, actually, in my opinion, it could be both, because so in general we know that and then the pathophysiology of FPAF, what it shows that the comorbidities can have a role in create systemic inflammation that can then lead to microvascular dysfunction and then to the symptoms of FPAF. And I think that as we see in chapter 2 that in all these different conditions, the platelets are aberrant, and we know that platelets can have, they can be influenced by the external environment, so they can be activated if there is like an imbalance between the priming factors in the circulation, but at the same time we know that platelets can influence the environment. So I think that if these platelets are...
Sorry to interrupt again, but for example, in diabetes, we know that platelets are, not functioning very well, but this is, do you think this is rather due to the diabetic state, or is it due to the platelet itself?
It's both, because we know that...
In other ways, when you treat diabetes, will you also improve platelet function?
Yeah, I mean, we do know that all the different condition linked to diabetes, like includes glucose and also insulin resistance, and also often these diabetic patients are also obese, and we know that the adipose tissues can release cytokines that can get this platelet activated, but we know that platelets also release in their granules, they release cytokines, and they can contribute to further inflammation. So there is a sort of dual aspect in this, bidirectional aspect in this. And we also know that some of these patients have a reduced response to antiplatelet medications, and maybe this is due to this platelet phenotypes that is hyperactive, and this is something that is in...
I agree totally. So my next question is, because I'm, as mentioned, I did a lot of research in patients with acute coronary syndromes and antiplatelet therapy. If you have a patient with HFpEF and you have this knowledge about platelet dysfunction, would you treat this patient with an anticoagulant or an antiplatelet agent? What do you prefer?
Yeah, well, I think that it's still probably still too early to what we know about the platelets. I mean, it's pretty preliminary, but I think that with anticoagulants will make sense if there is a presence of also atrial fibrillation, which is something that can increase the risk of thromboembolism. But I think that targeting platelets, especially in their interaction with endothelium, for example, would be interesting. So I will bet on that.
Yeah, for example, if I have one more minute, is this Prolector?
Very short, yeah.
Very short. For example, this you mentioned the less susceptibility of diabetes for aspirin, for example. Do you recommend diabetes patients to be treated with higher doses of aspirin because of this platelet dysfunction?
Yeah, I mean, could be, but I still think that, I mean, there is a, I think that also the antiplatelet medications are, I mean, there is research in that direction to improve also antiplatelet medications and their action. And I think that that is important because now it's more like the aspirin or the medication that are treating, they are targeting the receptor on platelets. It's, I think that also targeting different receptor or in the communication between platelet and other cells will be most effective. So there is room for improvement there.
Yeah, thank you very much for. I'm very happy with the answers and give the word back to the prorector. Thank you.
Thank you, Professor van het Hoof. We now continue with Professor Koene, Professor of Vascular Inflammation and Thrombosis at Maastricht University.
Thank you, Ms. Prorector. So, dear candidate, I would like to take the occasion to very briefly congratulate you and your team with this great result, which I have read with interest, and I think it's very well written and informative. So today I decided to be predictable, and I will focus my questions on Chapter 3, as you probably expected. And Chapter 3, the title starts with the word thromboinflammation. So my first question is, how would you define thromboinflammation, personally?
So, hi, listener opponent. Thank you for your compliment and your question. So, yeah, so thromboinflammation. Yeah, I would start saying that there is still quite a debate on what thromboinflammation is. Actually, I think that the idea around thromboinflammation is that there are different players, which are platelets, neutrophils, and coagulation, and they are all somehow dysfunctional, and then can lead to they can lead to thrombus formation, and so. Yeah, and this was, I think that, yeah, we don't see increased thrombosis in these patients, but I think that all these players are involved in FPF, and this is why it starts with the word thromboinflammation. But probably I would say that also using more like the mix between immuno and hemostatic function mix will be probably also another possible word to define this word.
Yeah, because you actually already answered in your answer. My follow-up question is that you don't happen to see thrombosis in a stricter sense. So I'll skip that, but I have enough questions for you, don't worry. So how would you distinguish this inflammatory state that you see with the inflammatory state that people witness in aged individuals? So where is the difference? How much of the information that you see is related to aging? Can you estimate that from the data that you have?
Yeah, I mean, it's known that there is inflammation linked to age, the so-called inflammation, I think. But I think that with FF, it's add an additional layers of complexity because it's not that just age. I think that there are differences because there are more players that need to be taken into account because there is all the comorbidities going, but also the dysfunctional endothelium, the impaired release of nitric oxygen, for example, and all these different contexts create, I think, a difference between inflammation that you have, like with aging, and this particular context that is also links maybe to more, it's seen more in females, for example. So there are more players in
Okay.
it.
Can you get this information from the reference values? Because of course you have an aged control population and you have reference values sort of dotted in your figures. I was quite surprised that these were quite different than the values you saw in your subjects. So how could you explain that?
Yeah, so for the measurement of the markers in plasma, we measure the difference between the patients and the controls, and then as there is no a reference value, for example, for platelets and neutrophils, endothelial markers, that is considered as a reference, standard reference. We also wanted to add an additional measurement, so we measure these markers also in a pool of plasma from healthy volunteers that were not matched as age for the age with the patients. And indeed, we did see a difference because there was, for example, for platelet marker, we see a more similar activation status in patients compared to controls, but a difference between the healthy volunteer. So, and this could maybe link to the idea, okay, this is maybe linked to age, only age, but actually in the platelet functional measurement, we do see a difference between the patients and the controls, and also when we correct for the age in the linear regression models, we still have significance. So. I think.
I had a teeny question, but I'll skip it and give the word back to the prorector. Thank you.
Maybe you can ask it during the reception. The next opponent is Dr. Eringa, Associate Professor of Microvascular Function and Dysfunction in Prediabetes from Amsterdam Medical Center and Maastricht University.
Thank you, Prorector. Dear candidate, I want to congratulate you with your beautiful thesis that you have constructed during the past years, and my congratulations extend to your promoter and co-promoter. So beautiful work, very hypothesis generating, and a lot of intriguing information in there, especially for someone who is not familiar with platelet function as I am. So, and HFpEF is also a new field for me, so maybe you can enlighten me a little bit based on your lay presentation, because you showed two pictures of the heart of a healthy person and a HFpEF patient that looked exactly the same. So I find that hard to imagine. So in what sense do they differ? And if I look at your cover, there's only one heart, so which I've been looking at for the past 50 minutes or so. So are we looking at a healthy heart or a HFpEF heart?
Esteemed opponent, thank you for your question and your compliment. Actually, the heart in my cover is a healthy heart because it was just easily, I guess, and it's also actually hard work from a teamwork between my sister and Max in the audience, of which I'm really proud of, but it's not my hard work.
Okay, great. So I'm going to interrupt you because I only have 6 minutes and we have a lot of ground to cover.
Yeah.
So now we're going to turn this healthy heart into a HFpEF heart. So what should we change to the cover image to make this a HFpEF heart? So what would be the key change from healthy to HFpEF?
So I think that the main change would be the stiffness of the left ventricle. That I think is the main difference.
Okay,
and,
so I can see that.
Yeah.
And so what would be the driving force for this stiffening? Because you mentioned the influence of comorbidities. We all know that HFpEF is a very complex disease, and you alluded to that. And page 54, with the characterization of the HFpEF patients, shows this complexity. It shows all the comorbidities. So which comorbidity is really most important for this HFpEF phenotype and the stiffening?
I think that one important one is hypertension, because the high pressure overload can then lead to cardiac hypertrophy, and then this leads to cardiac remodeling and to the stiffness of the left ventricle. So I think that that's just important.
I agree with that, but the blood pressure is actually the same in your control group, in your HFpEF group. So you happen to know the comorbidity that is very different between the two groups?
Actually, the controls are. I mean, it's because it's also the diagnosis of FF is not so easy. I mean, even for the clinicians, they have like a bunch of different informations that they need to say, okay, this patient is FF and this is not FF. This is why also all the controls went through echographical measurements, and also, for example, there are now these scores that combining the measurements in the blood, like for example the natriuretic peptides, and also other markers of cardiac stress, but also
I hoped you would enter my comfort zone here, because the HFpEF patients are obese. So they have a BMI of 30, whereas the lean controls, or the healthy controls, are, I think, at 27, which is not completely lean, but it's more or less okay. So to my great joy, you have dedicated 2 full pages of your thesis to theorizing on adipokines, so products of fat tissue in platelet function. So.
Of course. That's of course also another important point. Absolutely.
So which adipokine would be important in influencing platelet function?
I think leptin is important. In general, what is told is that these chemokines can bind to the receptor of platelets and induce increased platelet activation.
Can I ask one more tiny question, or do I have to? I see a no. So we'll continue the discussion over dinner, I suppose. So I'll happy to give the word back to the pro-rector.
Yeah, I'd like to give enough time to the last opponent. Thank you very much, Dr. Ehringa. The last opponent is Dr. Van den Akker, Assistant Professor of Regenerative and Reconstructive Medicine of Vascular Tissue Engineering, Maastricht University.
Thank you, Pro-Rector, and dear candidate, I'd also like to congratulate you and your promoters on this beautiful thesis on platelets and their role in HFpEF. Today, what I would like to do is we're hopping from comfort zone to comfort zone. I'd like to drag you into my comfort zone, and I'd like to discuss some key concepts on your work combined with literature, and I'm focusing on the interplay between blood platelets and vascular integrity. There are different places in your thesis where you point out that, I guess, also just during normal homeostasis, blood platelets are important in vascular integrity. Can you elaborate on that?
Esteemed opponents, thank you for your compliments and your question. Among all the different functions of platelets, I think that the old interaction between platelets and endothelial it's an important one. And actually, there is like a sort of dual role of platelets. So there is more role linked to the integrity of the endothelium, and this is linked also, for example, to the molecules that platelets are carriers in their granules. Like, for example, they contain growth factors, but also lipids. And it's also known that platelets are involved, for example, in angiogenesis. And this is can contribute to the integrity of the vessels. But actually, we also do know that in general, platelets and endothelial cells can interact through also a direct interaction. They can interact to different receptors, like, for example...
they can or they will?
I mean, it depends on the context. I mean, in the context of, for example, an inflamed endothelium that, for example, is expressing more p-selectins or ICAM or VCAM on the surface, platelet can bind these receptors through the, for example, the PGL. Sorry, p-selectin receptor. They can also.
But then you're talking about specifically an inflamed state, right? An inflammatory status of the endothelial cells. What would be the same process, or would there be comparable processes in healthy endothelium? Just let's say normal vascular integrity.
I mean, in general, when the endothelium is healthy, so there is not like an injury. One of the main aim of the endothelium is to keep the platelets inactive, because of course they don't want unwanted thrombi. So the endothelium is able to release molecules, like for example nitric oxygen or prostaglandin, that can actually inhibit platelet function. And this is really important because, otherwise you can have, like, of course, uncontrolled platelet formation. And I think it's important because, for example, in FPEF context, the role of nitric oxygen it's pretty important because there is.
And could you I mean, in this perspective, whether cause or consequence, it is the interaction of the platelet with the endothelial cells that either induces or drives forward anyway this HFpEF condition. Could you also consider vascular integrity beyond the endothelial cell? For example, in the coronary vasculature, would platelets also have an effect on other components of the vessels?
I think so. It's once exposed. I think so. I think that in this context, I think it's becoming way more important to investigate this relationship between platelets and endothelial cells, but there is like a large knowledge gap in this moment in the field, and I think it's a.
But would you hypothesize? If you don't know it, we can discuss on it. So you have a vessel, a coronary vessel, for example, coronary capillary. You've got your platelets. There's interaction. What more is a vessel than endothelial cells? And how would platelets have an effect on the rest of the vasculature besides the endothelial cells?
I think that's, I mean, like, for example, when there is, like, in general, vast injuries, and then when there is an exposure of collagen, and platelet can bind to collagen, it's one of the, main, activation and adhesion pathways in platelets.
So it would bind other cells than endothelial cells or other parts of the vessel besides endothelial cells when you lose vascular integrity. Would that be correct to say?
Yeah, I think so, yeah.
And what kind of, next to collagen, would you consider other cell types that are located there, involved, and how would they be a player in this complex cascade?
I think that, for example, most muscle cells and other cells can be involved, but however, of course, the endothelial is the first layer, and I think it's one of the things that should be.
Giorgia d'Italia, the time appointed for defending your thesis has passed, and the degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return in this room.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Southside, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like, my goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 10 miles in my rearview mirror. I know what it felt like, my goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like, my goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and die. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Giorgia d'Italia, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Kosemans is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Dear Giorgia, do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Giorgia d'Italia, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
Many congratulations, Giorgia.
Dear Giorgia, dear Dr. d'Italia, it's a true honor to be the first one to congratulate you on obtaining your PhD title. I would also very much like to congratulate your parents, your sister, your partner, other family members and friends present here and online. You did a great job during the defense. I'm very proud of you. You really took the stage and you were very enthusiastic and you answered questions very well. I would like to also give a special word of thanks to all the committee members present here and online for their time and effort in assessing the thesis of Giorgia and for being present today. In particular, I would like to thank the members online, Dr. Gianni Guidetti and Ea Lecone, for their presence. I will be delivering the laudatio together with your other promoter, Professor Blanche Schroen, and I will start with how it all started. It was actually May 2019. At that time, I received an email from Dr. Gianni Guidetti and Ilaria Conobio, and they told me, we have a very brilliant student that has applied for an Erasmus internship to go abroad. And Gianni and Ilaria, they are located in Pavia, Italy, and they're also fellow platelet scientists like me. And they told me her name is Giorgia d'Italia, and they described you as being very enthusiastic, diligent, curious, and precise. And I think this is a very good description of your qualities. And that's also how we saw you working the past 4 years, and also how we saw you today, enthusiastic and very knowledgeable. So I answered that I would love to host you, and you joined us in March 2020. And this was the time when the COVID pandemic just started. I remembered I received an email from HR, and they asked me, well, is it wise to have Giorgia coming over? And I said yes, because I didn't know what to expect. And after 1 day, we entered the lockdown. So we could only meet you for 1 day, and then the labs were closed. You stayed in a guest house in Maastricht, and the guest house became more and more empty over weeks. But you stayed, and you persevered. And I think perseverance is another strength of you, because you don't give up. Luckily, you were able to do lab work the final few months of your internship. And it also paid off in an unexpected way, because you met your partner during this time in Maastricht. Around the same time, Blanche Groen and we obtained funding from Karen to appoint a PhD student to study platelets and heart failure with preserved ejection fraction, in close collaboration with Dr. Vanessa van Empel, cardiologist here in Maastricht, and her team. We approached you, you accepted the position and started in January 2021. And again, this was a difficult time because of the COVID pandemic, and traveling by plane was difficult to Maastricht. But again, you didn't give up, you are quite determined, and you traveled by car from Genoa, Italy, to Maastricht to start the journey of your PhD. Over the years, your work has been instrumental, not just for your PhD, but also in helping us to secure a prestigious grant from the Dutch, British, and German heart foundations. You mostly work quietly and operate in the background, but you do take the stage when there's music on the dancing floor, but also here today. Very well done. I'm proud of you, Giorgia, not only with regard to your scientific achievements, but also by your personal growth over the last years. Keep bringing up your unique strength to the scientific world and the world in general. And with that, I hand over to Blanche.
Thank you. Well, Giorgia, also from me, congratulations, and also to your family and partner. So you can be proud. I'm also proud. The word proud is going to be mentioned many times. So I'll tell a little bit about when you joined us for your PhD, because that's when I met you. And you were so full of excitement. And you still are. We've witnessed that today. There is so much energy in you, and you are so generous with giving it to your work and to other people. So that's something that stood out for me. Your optimistic nature and your cheerfulness also stood out, and it was challenging. The PhD was challenging. The first thing that you and actually the whole team needed to tackle was our language. Not English, Dutch, or Italian, but the expert language. With your project, we wanted to bridge several domains in biology: platelet biology, biochemistry, which are Judith's domains, but also the domains of molecular genetics and physiology and cardiology, which are more my domains, and where Vanessa also came in. I remember vividly how you taught me about the coagulation cascade and why we stimulated the platelets in the way we did in the lab. You were really good at it, and often we supervisors forget that we don't know everything. And it really helped me to be the dummy. In that way, you helped the project as well, because our common understanding of that key experiment resulted in an improved setup. I think we grew together, and that is one of the reasons that made me very proud of the end results that we have just witnessed through your excellent defense. The second hurdle was a bit more personal. You needed to tackle Judith and me. Two different characters in two different cultures. Indeed, both Judith and me, but also the departments we are in, are so different. For all sorts of reasons, of course. And I'm very curious what stands out for you the most, and maybe we will hear a bit more about that tonight. What stood out for me is that we put you through a considerable challenge by asking you to work in two different departments, two different labs. And luckily there was the great support of the technicians in both departments, Simone in the audience, but also Wouter, Rick and Chantal. And with their help you did the best job you could. As also proven by the diversity of the topics and techniques in your thesis. The real surprise came towards the end, at least for me, when you turned out to have a talent for statistics. Lloyd can definitely judge that much better than we can, at least you did and I can, but the output you produced under his excellent guidance was key. Also very important, we saw you lighten up. You grew in confidence with every table you produced, with every statistical test you did. It was a pleasure to witness this transition in you. So now we are here. In this moment of celebration, I want to congratulate you with this achievement and include everyone as I did before. I heard you have a job as well and can continue to pursue your dream of being a scientist. I wish you well and do stay in touch.
Dear Dr. D'Italia, also on behalf of Maastricht University, I would like to congratulate you with the degree you have obtained. I also congratulate your family, friends and colleagues here or online with this achievement, because it really is an achievement. I'd like to congratulate your supervisor with this very good PhD defense, PhD trajectory, successful. I'd like to thank the committee members present here and present online, especially our guests from outside Maastricht University, Dr. Lahakonen and Dr. Giudetti. Thank you very much for being present during this ceremony and investing your time in preparing yourself. And for all the committee members, I really enjoyed the lively discussions we had during this PhD defense. I can tell you that the committee is very happy with the way you defended your thesis. I hope you yourself are also happy with the way it went. I wish you a lot of success in your future career. I understand you go back to Italy. Yeah. And, well, enjoy the next couple of years with the studies you're going to do then. And I hope you have an enjoyable rest of the day. And with this, I close this academic ceremony. We are going to stay here to make some pictures. I would like to ask the audience, except the family, to go to the rafter for the
Reception.
Reception. Yeah. Couldn't find the word. For the reception.